Hello, my name is Jungmin Lee. I am part of Urban Ecosystem FRI Lab, Team Ruth, researching in ICP-MS Lab. I'll be introducing what I am researching in this ICP-MS Lab, what instrument is used, how we analyze the data, and where the results can be used. The research is to detect concentration of heavy metals such as lead, copper, chromium, zinc, silver, cadmium, and arsenic in the creek water in UT Austin campus. I will collect sample water from 5 different spots from the creek, dilute them, put them in the ICP-MS in order to detect concentration of heavy metals. So, what is ICP-MS and how does it work? ICP-MS is an instrument used to detect heavy metals. It is a mass spectrometry that uses plasma to ionize and atomize the sample and create polyatomic ions that can be detected in the later stage. Plasma is generated from 3 electrodes, all pointing the same direction at one end. So basically, ICP-MS ionizes and atomizes the sample in it to detect what is in the sample. This allows us to detect what heavy metals are in the water and analyze it further to calculate the concentration of detected metals. So how do we dilute these sample waters? After collecting 5 sample water, I will filter those water and dilute them using nitric acid. I would first mix 200 microliters of sample water with 800 microliter of nitric acid. Using the equation as listed, concentration times volume equals another concentration times volume. The list of volumes are shown on the first half for the nitric acid. I will repeat this process for each volume. Then, to prepare a calibration curve, I will use standard solution of lead nitrate. I will use the same equation and do the same thing, but with the different volumes. Those volumes are listed under Prepare Old Standard Calibration Curve. So, how do we analyze this data from ICP-MS? I will show you an example data that is given by ICP-MS. So this is the ICP-MS data that is given to us when it is done. Each column represents each metal that is detected from the sample. The green row, or the green fonts, can be ignored, and these orange fonts are used to create calibration curve and an equation for that curve. Data that will be analyzed by us are these data in black fonts. I would copy and paste average intensity and standard intensity listed right here on a new separate sheet. As an example, I will show you analyzed data of Cu. So, as you can see, those average intensity and standard intensity are copied here. I will plug in average intensity for Y that in the equation from the calibration curve and solve for X, which will be my calculated ppb. Milligrams per liter is calculated by multiplying concentration over 1,000. Concentration is the number here, and so in this case, 1,000 over 1,000 will be 1. So milligrams per liter equals to the calculated ppb. Standard deviation is calculated by dividing standard intensity by average intensity. Real standard deviation is calculated by milligrams per liter times standard intensity. This is used as error bars in the graph right here. This whole process of collecting data and analyzing these data will be done for each site, for each metals. So how can these results can be used? Detecting metals from the creek water is significant since those concentrations can affect plants and bacteria that are later eaten by other predators. If there is any concerning concentration of heavy metals, can be sent to environmental organizations, water facility, or even to the local government which is where experts can find answers to the problem. So what can we do for future labs? The lab is planning to research about biofilms. It will help to collect further data with details since metals can be clogged and attached in these biofilms. I will be able to observe higher concentration of heavy metals. Even some more types of heavy metals can be detected since biofilms can have higher concentration of heavy metals that couldn't be detected earlier. That is it for the introduction of the ICP-MS lab. Thank you.